Hello and welcome to Better Biopharma. I'm your host, Tyler Menichiello, and I'm joined today by Dr. Fiona Elwood, the Vice President and Disease Area Stronghold Leader for Neurodegeneration at Johnson & Johnson, where she leads the scientific and strategic direction of J&J's modality-agnostic neurodegeneration pipeline. Dr. Elwood, thank you so much for being here today.
Thanks. It's great to be here, Tyler.
So, for our folks at home, would you please start us off by describing your role as the disease area stronghold leader for neurodegeneration at J&J and how this connects across functions at the company with teams such as discovery, development, and how it touches commercial strategy as well?
Great. Thanks. Yes, because I love my role and it's quite unique in the industry, I think. So as you said, I'm responsible for the science and strategy of our neurodegeneration portfolio. And at J&J, we are organized in end-to-end therapeutic areas. So we have discovery, early development, late development, all within one neuroscience organization. And that's really attractive to me because, I came from the discovery organization, worked in early development, and I've seen, there's challenges that you have if discovery is divorced from early development or if early development is separate from late development. But in J&J, it's end-to-end, all under one head of neuroscience. And then I have the neurodegeneration third of the portfolio. So we work on neuropsychiatry, on ophthalmology, and neurodegeneration. And within that, we work primarily on Alzheimer's disease, but also on Parkinson's disease and some autoantibody-driven diseases as well.
That's terrific. Thank you. And so I want to dig into J&J's approach here. Um, and I'm curious, we talked about this disease-specific approach, but can you tell our audience a little bit about why J&J takes this disease-focused approach, and in many cases, modality-agnostic?
I think it's really organized around the patient and that focus around the patient, their experience of the disease, that the unmet medical needs, and really what they need and what their journey is as a patient for a particular disease. And essentially, the strategy is that if we can really understand that disease very deeply, the patient's needs, the science of disease, how care is administered for this type of patient, the more deeply we can understand that, the better we can bring solutions, therapeutic solutions for that patient and for the whole spectrum of disease that each patient might experience. So what does that look like for us? So for example, in Alzheimer's disease, we have programs that target from the very earliest stages to the very latest stages of the disease. We're looking at secondary prevention. So when patients already have Alzheimer's pathology in their brains, but they haven't yet moved to the phase of disease where they're experiencing cognitive decline. We have programs targeted at that group of patients to prevent them from ever developing cognitive symptoms in the first place. We have programs targeted at early Alzheimer's disease patients. So people who are already starting to experience mild cognitive impairment due to Alzheimer's or early Alzheimer's disease. And then we have programs targeting later stages of disease as well, where patients often develop challenging psychiatric symptoms, either of agitation, aggression, psychosis, and we have programs for those patients as well.
That's great. I do want to speak a little bit or have you speak on Alzheimer's in a bit, but I was curious, coming from your background, how does J&J's organizational model kind of fly in the face of your own experience at other pharma companies, and especially how does it compare to the traditional kind of more siloed structure and move towards this integrated development organization?
Yeah, absolutely. Different people at different stages of disease have great insight and great expertise around, their focus. And that can be great for developing wonderful molecules or thinking about efficient early development or thinking about, sort of broad patient engaged registration strategies. But often when those groups are separate, they are either competing with resources for very different types of disease, where that sort of format, what's known, the models, the state of biomarkers, the types of clinical development you might do could be very different. And also, when you hand off from one group to the next group, there can be a real gap in that understanding of the molecule and of the disease and of the patient, which leads to delays or misalignment, potentially, of that development path. The last thing we want to do is delay the advance of molecules to the patients who need them.
Yeah. Thank you. For our audience, why don't we have you talk a little bit about the type of modalities and molecules or assets in your pipeline and the indication specifically you're working on right now. They hear us keep talking about Alzheimer's. So why don't you talk a little bit about the disease areas we're looking at and the lead candidates?
Yeah, absolutely.
Yeah.
And you said, I mentioned just earlier that we have, a portfolio that really targets kind of end to end of the disease trajectory. We are also, as you said, modality agnostic. At the earliest stage, we're looking at an active immunotherapy. So this is, injected intramuscularly into patients who have Alzheimer's pathology. And the goal is to generate an antibody repertoire that could intercept the disease pathology and the spread of the disease pathology and stop patients from ever experiencing the onset of cognitive decline. So active immunotherapy, number one. And that's really targeted towards that stage of disease as well. These would be active people who would be sort of still fully engaged in their lives, probably not yet retired, but, on the trajectory to developing cognitive decline. So they have very busy lives, lots going on, and they want to intercept their disease safely, effectively, and without a large burden on the rest of their lives. Short intramuscular injection. The next is posdinemab, as we talked about. So that's the monoclonal antibody targeting early Alzheimer's disease. This is a monoclonal antibody. We can infuse high levels of this antibody, but it's given IV. So it's also required to be delivered in the context of a clinic with additional professionals providing support and care for patients who already have started that descent into cognitive decline and symptomatic Alzheimer's disease. And then finally, we have the molecules for agitation and psychosis in Alzheimer's disease. And actually, as I mentioned before, with J&J, we have big psychiatry group as well as our neurodegeneration group. And this comes from really nice cross-disease area collaboration between the two different groups. And here we're testing a molecule, ITCI 1284, where we're testing that molecule. And it's a small molecule, which, fits well with the sort of pharmacology and with the therapies that are often administered to these later stage patients.
Yeah. Thank you. So how is it overseeing so many different, like a modality agnostic organization like this? What was that like getting used to and just kind of learning this organizational structure of J&J? And you mentioned that cross-functional collaboration there with psychology division.
With the psychiatry division, yes.
Psychiatry, yeah.
You know, um, uh, we have fantastic functions at each level, and whether that's at the modality level, at stage of development, and as you can imagine, with a model like this, we have strong collaboration between the different groups. So I work really closely with the experts in discovery, development, biomarkers, as well as the experts in our kind of big central groups at J&J, who are just fantastic. And really driving that modality agnostic platform, experts in all different types of biological molecules, as well as novel, small molecule modalities that really enable us to go after the best target in the best possible way. So lots of collaboration, lots of communication, and I think a mutual respect for what the different team members bring to the table, for really being able to advance that pipeline.
Yeah. Thank you. Just to dive more, double down on the modality agnostic angle here. I'm curious, how does J&J match disease targets with the appropriate or optimal therapeutic modalities? Like, how do you evaluate what modality is ideal? And what does it really mean to be modality agnostic, and how does that mindset shape the
Yeah.
program development?
I think it comes patient first. What does that patient need? Target. What are the different ways that we could address or modify that target? And then we think about what modality would we bring forward. I think it's potentially enabled by the scale of J&J. We're a really large organization. So we are able to either bring internally or through partnerships a whole range of different types of molecule to play in the diseases that we're focused on and the targets that we're focused on. And, both the great sort of internal discovery engine that we built, as well as our, long history in engaging with the external world, really mean that we can take our pick. We're not limited by the type of molecule that we would propose for the target and the patient, the needs of the target that we think we need to go after, and the patient and what they need.
Yeah, that's great. Thank you. And so I did want to talk a bit about Alzheimer's because when we last spoke, we were talking about the changing treatment paradigm of Alzheimer's. And I'm curious, looking at the early days of your career, how would you say the way the industry as a whole is thinking about Alzheimer's has changed?
Oh my
And
gosh, it's so exciting being at this stage for AD. I think I've been working in Alzheimer's for 25 years. And, I would say for the first 20 of those years, we were in our natural history era.
mm,
We were thinking about really trying to understand the biology of how the disease progressed. That's actually how I ended up working in Alzheimer's disease. I was really interested in the brain, in neuroscience, and Alzheimer's was the first place where the field was making traction in understanding human genetics, human pathology, and being able to link that to the biology of the disease. So, we spent a long period of time in our industry with some clinical trials and some failed, failed clinical trials where we as a field have learned huge amounts from every single clinical trial that's been tried. Um, but we were really trying to understand the natural history of disease, and there were some really fantastic breakthroughs from in the biomarker space, in the imaging space, amyloid PET, tau PET, in the biomarker space, fluid biomarkers, first in the CSF and now in the plasma, where you can actually do a blood test and measure particular proteins that come from the brain that correlate and that tell you the status of the Alzheimer's disease pathology in the brain. And then even beyond that, so things like the GNPC, Global Neurodegeneration Proteomics Consortia, which is like a really broad-based consortia set up by J&J and Gates Foundation to look at 7,000 protein analytes in the plasma of tens of thousands of patients who have neurodegeneration. Or UK Biobank, or you name it, all of these huge public-private partnerships to really understand that biology of disease. That 20 years set up our understanding of disease progression in Alzheimer's disease, in mechanisms of disease, even today moving to different subtypes of disease. And then now we're in a place where we have, biomarkers where we can measure and stage patients. We have therapies for that are starting to be able to treat and slow the progression of disease that are available for patients. We have healthcare systems that are adapting to this new sort of era, the treatment era of Alzheimer's disease, where we're able to actually slow the progression of disease. Just to start, there's so much more need for patients for safe, effective, convenient therapies. But after that 20 years of understanding disease, not even really being able to measure disease in real life, only at postmortem, to this point where we have biomarkers, we can identify patients, and we're starting to treat patients. That's such a change. It's super exciting to be able to work in this field.
yeah. So I'm curious, what exact biomarkers come to your mind? I mean, I'm not in any way an Alzheimer's expert or have ever worked in neuro, but I know the hallmark of Alzheimer's disease are the tau proteins and the beta plaques or amyloid beta plaques. So, and I did know that previously you couldn't see those in the brain until post mortem. So, are we able to just grab these out of spinal taps, or what kind of biomarkers are in your mind when you're saying that we found out these biomarkers, we've improved diagnostics? Like, walk me through a little bit of.
Sure. It's even better than that. So we can measure the amyloid and the tau in the brains of living patients using PET, positron electron tomography, by a method of, an infusion and a scan where we can measure and image the amyloid and the tau PET in patients' brains. Amyloid is important. That's the target of the current disease-modifying therapies. And, that sort of really set up and helped establish this staging system for disease. Tau PET is really important because the level of tau as an input is important, but where the tau is in the brain is also really important too. As the tau spreads through the brain, so that correlates with the brain circuits that are disrupted and the symptoms that the patients experience. And there's a much tighter correlation between the tau pathology in the brain, where that is, and the symptoms and the progression of cognitive decline that patients experience. So both of those two imaging technologies have become really, really important. We're actually using that tau PET to measure whether our monoclonal antibody, posdinemab, can block the spread of tau from one brain region to another using some advanced analytics and how we measure that distribution of tau through the brain. But the other type of biomarkers that you mentioned are actually plasma biomarkers, so blood-based biomarkers. And you may have seen there was just regulatory approval of the first blood-based biomarker for the diagnosis or confirmation of diagnosis of Alzheimer's disease. And there we can measure different fragments, either of amyloid or, most importantly, of tau, which are the most sensitive measures of Alzheimer's disease pathology in a patient's brain. So you can now do that just from a blood test. And I'm super proud to work at J&J because actually some of the foundational science for that was done from our labs, and we will develop one of the first blood tests for AD, our phospho-217 assay, and use that, have been using that for several years in our clinical trials.
Wow, that's terrific. Thank you. Yeah, I think the technology around diagnostics and more effective diagnostics is an underrated, maybe not, but in my opinion, it's an underrated kind of
Yeah.
hallmark of the industry that doesn't get enough love, because I just think it's incredible to be able to see inside the body with such precision and detail that it was unheard of even a decade ago.
Absolutely. And what's important for neurodegeneration is to be able to identify and treat patients as early as possible. We don't want to catch patients who are at, moderate or late stages of Alzheimer's disease. We want to catch them even before the onset of cognitive decline. You can have amyloid and tau in your brain decades before you start to see a change in cognition, because there's that delay in neurodegeneration and then the onset of symptoms. We have capacity in our brain to deal with some of those early impairments. But we want to capture patients at that earliest stage so that we can stop the progression of disease at the earliest stage when the brain is least damaged. Because, we think it's going to be easier to be able to identify people and stop the progression of disease rather than try and grow back a brain which, hasn't been growing since we were much younger.
Yeah. Yeah, to use a probably poor analogy, it's easier to stop a leaking pipe than to just rebuild the whole
Build the
house,
whole system.
right?
Yeah, exactly.
Right.
Exactly. So you need those biomarkers. You need convenient early access biomarkers in order to be able to do that.
Yeah.
Blood tests that your PCP can order, for example.
Yeah. And I was going to ask you about how this understanding of the biology behind the disease that we are afforded through these kind of um, biomarkers and tests that you're alluding to. How does that understanding and patient segmentation, as a
Yep.
take pulled from that, how does that affect or guide the decision making across J&J's pipeline?
We really have this precision approach in our neuroscience department. So we're focused on the right drug for the right patient at the right time. Often in Alzheimer's disease, that means we're focused on the stage of disease. For our active immunotherapy and tau targeting monoclonal antibody, our goal is to stop the spread of disease, stop the spread of pathology, tau pathology from one brain region to the next. So we're enrolling patients who have that specific level of tau pathology. And that's how we design our clinical trials and think about how we're developing these molecules for patients. Beyond that, though, Alzheimer's disease patients have this stereotypic progression of disease, but they're heterogeneous too. And different patients can experience different co-pathologies, whether that's cerebrovascular co-pathology or aggregates of other proteins like TDP-43 or alpha-synuclein. Or they can also have, changes in sort of that underlying biology propensity to develop disease. And we're starting to see that through some of these global proteomics consortia that I was mentioning before, the GNPC and others, where we can identify different subtypes of disease. And then our goal is to be able to target the subtypes of disease as well as the different stages of disease to really afford greater benefit to patients and to target the right therapy to the patient who's going to benefit from it most.
Yeah. And do any of these early decisions around disease models or patient subtypes and ultimately modalities, does that create any challenges downstream for process development or scaling up? Like, I'm curious, I suppose, how in J&J's ecosystem, if there's any, is there crosstalk upstream and downstream versus, as opposed to, subtypes and modalities and just ideal means of
Yeah, I mean, I
targeting?
think that end-to-end structure really enables that crosstalk up and down the ecosystem, by having sort of one view on the biology, the early development, the late development, the patient journey, the clinical experience, and having all of those experts in those different areas talk to each other under the umbrella of our neurodegeneration and our Alzheimer's portfolio. That really enables, bi-directional communication. Specifically when it comes to the stages or the subtypes of disease, I think, I just kind of refer to what I said earlier about choosing the right modality for the patient and for the target. And that we can do that when we think about the full picture of treatments for patients in the future. So, for example, in Alzheimer's disease, it's likely that we're going to need combination therapy for Alzheimer's disease patients. I'm going to go out on a limb and say we don't want to be treating an elderly person with multiple different infusions of different antibodies for an extended period of time. And so when we're thinking about, the whole treatment of the whole patient and that combination approach, we think about how those different modalities could be combined in the future and the regulatory development, safety aspects of those as well.
Yeah, that's great. Thank you. And I was curious if you could offer any advice to those listening in the audience who might be coming from smaller organizations that are looking to de-risk their early programs, but also bearing in mind development and manufacturing needs. How do you strike a balance there in terms of finding the most effective approach to treating a disease, but also bearing in mind the complicated developmentability, if that's even a word, of some of these drugs and the manufacturability of them?
Yeah, that's why we have the collaboration and we have forums where we really have the insights that come from, multiple different stages of the process who get to share their perspective and their insights throughout the drug discovery and development process. One thing maybe that's a little different about neurodegeneration is how long it takes for our clinical trials. We're talking about slowing the progression of slow diseases. And then we're talking about going even earlier and slowing the onset of symptoms in slowly progressing diseases. So we have clinical trials that are really, really long. When I look at my immunology colleagues and they say, oh, yeah, my proof of concept study, that's going to be 2 months, 3 months long. And I'm like, oh, my proof of concept study could be 4 years long.
Wow.
There's real differences in like the duration of our clinical trials. Um, and so, we always look to see how we can optimize the progression of those clinical trials and that clinical development plan. And that comes with staged investment at risk at different stages of development. So, the challenge that we often have in neurodegeneration is that we might be thinking about investing in scale up, not just for phase 3, but potentially for commercial, for launch, before we have all the data we would like because of the slowly progressing disease. Or the alternative is to push that decision out to later and then, extend the launch time for the molecule as well. So these are some of the trade-off decisions that we have to have and conversations that we have to have about, what level of investment we need to make at what time and how much data we have in order to really make a de-risk decision for some of these large investments.
Yeah. Understood. Thank you. I did want to ask you some more about Alzheimer's. In some conversations I've had, I won't name names, but there seems to be this emerging school of thought of Alzheimer's as an inflammatory disease. Um, and I'm curious where you stand on that because, like we were talking about before, these protein aggregates, the tau proteins, and the plaques, the amyloid plaques, are characteristic hallmarks of the disease. But I've heard them described more as a cause of inflammation, and if the inflammation itself is targeted, then that could have a therapeutic benefit. I'm curious, how do you feel about the school of thought, and where do you kind of sit in that?
Yeah, I think scientifically, there's some really interesting points there. So, hands down, when we look at Alzheimer's disease genetics, half of the SNPs, the mutations that increase the risk of developing disease, map to some of the inflammatory cells in the brain, to the microglia, which are like the macrophages in the brain. So clearly, there's a really important role for neuroinflammation, the innate immune system in the development of Alzheimer's disease. When we look at biomarkers and try and understand what correlates best or what predicts future cognitive impairment, the tau pathology is really what predicts future cognitive impairment best. Better than amyloid, better than inflammation, better than some of these other approaches. Um, and so we have this balance of, what's happening to initiate or start or drive some of the earliest stages of disease versus what really correlates or drives the development of disease once pathology has developed at the start of cognitive decline. There's no doubt that the immune system plays an important role in Alzheimer's disease. I think the questions are more around at what stage is it most relevant and most important in disease? And what would be the best way to target it? Because the immune system can play both beneficial and detrimental roles in disease. So that's the challenge for the drug hunter is to understand who's the patient, who are we targeting, who's most likely to benefit, and then also how can we do this in a way that is safe and efficacious. And I think when it comes to the neuroimmune axis, there's so much more that we have to learn in that area. Tau is definitely, amyloid is out in front of that, tau is out in front of that, but there's so much more we need to learn in the field as we continue to progress the science and our understanding of disease.
Yeah. And I'm curious to hear from you what are you most excited by, I should say, in the Alzheimer's treatment sphere and scope. Obviously, like even at J&J, there's a lot of different approaches, modality specific approaches, depending on, like we were talking about, the patient subtypes. What are you excited by in Alzheimer's? And yeah, I'll just leave it out like that.
Well, and thank you. Yeah, we've touched on a couple of those. So the biomarkers, the ability to identify patients, even identifying cognitively unimpaired people that they have the presence of amyloid and tau pathology in their brains. That is an absolute game changer, as we sort of discussed earlier in the conversation. Secondly, I'm super excited about our portfolio and some of these tau targeting therapies that we're advancing and that we're really at the forefront of. front and center being posdinemab, our monoclonal antibody that's fully enrolled, and that we're evaluating for the treatment of early Alzheimer's disease. I'm really intrigued to see the results of that study and see whether we can open up a new avenue for treatment for Alzheimer's disease patients. And then maybe third, it's this continuing, it's the scientist in me, the discovery scientist in me, that's this continued exploration and understanding of human disease, whether it's the neuroimmune axis, whether it's subtypes of disease, mitochondrial dysfunction, blood-brain barrier dysfunction, to help us understand more about the etiology of this disease, how it progresses, and think about new ways of targeting the disease and bringing even greater impact to patients as we go forward.
Yeah, that's great. Thank you. Well, Doctor Elwood, I know we're running a little bit short on time here, but I want to ask you the hallmark question that I use to wrap up every episode. I ask all the guests, and that is, in your opinion, looking at the industry as a whole and or your specific sphere and scope, what do you think we can do to better biopharma?
I love it. It's a great question. Um, I think the more we try and understand the insights from the patient, the experience of the patient, what they go through to get a diagnosis or to access care, the impact that having that care has on their lives, the more we can understand their full experience and bring that into every stage of the drug discovery, development, manufacturing process, the better solutions we will bring, the bigger impact we'll have to people. So I would say we can better biopharma by really being focused on having the patient at the center of everything we do.
Great. Thank you so much, Dr. Elwood. And thank you so much for tuning in to this episode of Better Biopharma, the official podcast of Bioprocess Online. Stay tuned for new episodes released every other Wednesday. We'll see you next time.